Okay, so in this video, we're going to be going over the link reaction. And also we're going to go over the Kreb cycle. Okay, so the Kreb cycle is also known as the citric acid cycle. Okay, all right, so now coming out of glycolysis, we have a pyruvate here, right? Because we know in glycolysis, we made electron carriers, we made a pyruvate. So here's our pyruvate. And that pyruvate is now going to move into the mitochondria. I'm going to just make a big kind of fat mitochondria here, okay? And it's when it moves into the mitochondria, to move that pyruvate into the mitochondria, a couple things are going to happen, all right? And this is called the link reaction. So that pyruvate is going to be decarboxylated. Carboxylation. Okay, so it's going to decarboxylate. So that means if we've lost a carbon, the final product that goes into the mitochondria is only going to have 2 carbons. Okay. The other thing that's going to happen is it's going to be oxidized, right? So we're going to have oxidation. So that means we're going to have the NAD plus turn into NADH plus H plus. Okay, so we've got decarboxylation, oxidation, and we're also going to add coenzyme A. So that's just an additional molecule. Okay, we're going to add that too. So our final product is going to be called acetyl-CoA. Okay, so there's our acetyl-CoA. Now, this is what is going to enter the Krebs cycle. Okay, and this acetyl-CoA is important because that's how it's going to attach to the first kind of cycle through. So this is going to be a cycle, right? We're going to move, this Krebs cycle goes around and around and around. And the first molecule that this coenzyme A is going to attach to is one you need to know. It's a 5-carbon sugar called oxalacetate. Okay. Oh, I'm sorry. It's a 4-carbon sugar. I take that back. So oxaloacetate. All right. Now, so this is a 4-carbon sugar. Okay. So when we attach the extra 2 carbon to it, right, we end up getting a 6-carbon sugar, okay? And coenzyme A goes away. That was just used to help attach it, okay? So we've taken a 2-carbon acetyl-CoA and added that to the oxalacetate to create a 6-carbon sugar called citrate, okay? So here's our 6-carbon sugar. Now, that citrate is going to go through a series of steps of decarboxylation and oxidation to create, the oxidation is going to create electron carriers, NADH, and the decarboxylation is going to result in a bunch of CO2. So first, we're going to get decarboxylation and oxidation, okay? So decarboxylation means we're going to take a carbon out of that, and if we take a carbon out of that, we're only going to have 5 carbons now, from 6 to 5, okay? And the other thing that's going to happen is we're going to have the oxidation happen, so we'll have an NAD plus, right? Go to NADH plus H plus, okay? And these guys we know are electron carriers, right? Plenty of energy. All right, and that's going to happen again, that same process. Okay, so we're going to have a decarboxylation, and we're going to have oxidation. Oops. NAD plus goes to NADH Okay, so decarboxylation and oxidation. Here's another NAD. Okay, H plus H plus. And of course, since it's decarboxylated again, now we just have a 4-carbon sugar, right? And so we can just call these 5-carbon sugar or 5-carbon acid 4-carbon sugar, okay? And now what happens next is there are going to be some reactions that result in the synthesis of ATP. So we're going to get this is substrate level, so there's enzymes involved, ADP, and we're going to go to ATP. And there's a phosphate that gets added to this somewhere in between. You don't need to worry about that. But here we have ATP synthesis, right? So here's our ATP, okay? And then on the way back, after that happens, we're going to actually have another oxidation, but that oxidation is going to be not with NAD, it's going to be with FAD. This is a different electron carrier. And the FAD will be reduced to FADH2, okay? So this is another electron carrier. It's just a different kind. And then, of course, that is still a 4-carbon because we haven't decarboxylated. And then the last step, we're going to end up doing one more oxidation. So, and this one is another NADH. Okay, so there's another oxidation. Okay, so, and when that happens, it finally becomes oxaloacetate again. All right, you need to know the terms acetyl-CoA. Acetyl-CoA attaches to oxaloacetate, a 4-carbon sugar. Okay, you need to know this term as well, acetyl-CoA and oxaloacetate. And what comes from that is citrate, a 6-carbon sugar. You should know that. Then it gets decarboxylated twice, and it's also oxidized twice. So that means this is going to lose electrons along the way, and we get electron carriers. Then ATP synthesis is going to happen. This is substrate level. Okay. And then we get oxidation happening again and again. The results from one pyruvate, right? So for one pyruvate, just for one single pyruvate, we're going to get 1, 2, 3 NADH plus H plus. We're going to get, let's do 1 FADH2. And we're going to get 1 ATP, okay? Now, this is for one pyruvate. So if that's for one pyruvate, if you were to go back to the original glucose molecule, you would multiply everything by 2 if you have the glucose, if you start with that glucose. Like if you were to say, how much comes out of one glucose molecule, you know, when you go through this process is you get 3. So that would be 6, 2, and 2, right? Okay, so the result for one pyruvate is 3, 1, and 1. But if we look at it at the glucose level, because you get 2 pyruvates for every glucose, is that would be 6, 2, and 2. Okay? All right.